samples from the diseased brain, and it's not in the samples from the normal brain. And this is exactly what we had been looking for. We and everybody else have been looking for some sort of a key, some kind of a molecule that we could say was in the diseased brain and not in the normal brain. And that really forced us to conclude that this protein is part of the agent, in fact, might be the only component of the agent.
This band of protein appeared to be the only difference between the diseased brains and the normal brains. They called it the Prp protein. Finding it didn't solve the mystery, though. It deepened it, because no one thought a protein could be an infectious agent. Apart from water, our bodies are composed largely of proteins. There are thousands of different kinds of proteins, aiding in everything from digestion to thinking. But proteins contain no genetic material, no nucleic acid which would allow them to reproduce. So how could a protein multiply and cause disease?
To the best of my knowledge, all infectious agents, all pathogens, require the participation of nucleic acid in order to multiply, to replicate. The idea, therefore, that replication could occur without nucleic acid is heresy.
We couldn't figure out how could a protein replicate if it doesn't have a nucleic acid.
The answer had been suggested decades earlier, but dismissed as too outlandish.
I started thinking about several years before, when I had been doing some literature review and looking in some journals in the library at a paper, and I was looking actually up one paper that was written about the nature of the scrapie agent, and when I went to photocopy it, on the second page of that paper, there was another paper, and it was by a mathematician named J.S. Griffith. And in this paper, he talks about self-replication and scrapie, which is one of these diseases. And he outlines 3 ways that a protein alone could replicate and cause disease. Now, he came up with the idea that you would have 2 different forms of the protein. One was abnormal, disease-causing, a rogue protein, if you will. And the other was a normal protein that would be in the cell or in the brain of a normal person. And the essential part of the theory was that the abnormal form of the protein could bind to the normal form and convert it, change its structure to make it an abnormal or a disease-causing protein. And now you can see that if one molecule binds to another molecule and converts it, now you have 2 molecules of abnormal protein, and 2 can bind to 2 more and have 4, and so on until you have thousands and millions of abnormal proteins in the brain, and pretty soon then you have disease. You have so much abnormal protein in the brain that it would cause disease and cause neurons to die. The most amazing thing was that the paper was published in 1967. He was a mathematician, wrote this one paper, never published anything again in scrapie. The insight was really very astounding that someone back then with so little information could see a way that a protein could be an infectious agent.
Since then, the idea has been taken up by many scientists, most notably Stanley Prusiner. He and others found that there are 2 types of Prp protein in the brain, just as Griffith predicted. One is a normal protein found in all mammals. The other is a disease-causing form of the protein, called a prion, a term coined by Prusiner. The prion is chemically identical to the original protein, but it has a different shape, which makes it so stable that it resists heat and disinfectants. It appears to have no genetic material, so radiation does not harm it. Once in its abnormal form, this new molecule seems to have the ability to corrupt any healthy PRP proteins that it comes into contact with, turning them into prions too. This is not replication, it's conversion.
It's a very strange observation that you have these two quite different forms of the same protein with quite different properties. One of them is a killer. If this protein is present in your brain, you're in serious trouble. The other one is a normal constituent of all our brains. And obviously understanding how one converts into the other, and in the longer term how to stop it, is a tremendous puzzle.
While scientists are still piecing that puzzle together, they have found that prions can link up in long, indestructible chains that accumulate in the brain. There, they kill cells, creating the holes that turn the brain to sponge. If correct, the prion or protein-only hypothesis would be revolutionary, a phenomenon unheard of before in biology.
I very strongly believe in the protein-only hypothesis.
Notice only the old people.
But it remains unproven and controversial. Some believe that prions are not the cause of disease, but the byproduct of it. Others think the infectious agent might turn out to be a virus or something similar, its genetic material hidden in a protective coat of protein.
The idea that it is an infectious protein is an exciting and curious idea, but is one which I do not accept.
This would be a novel biological mechanism. It's been difficult to accept because of that, because there's no precedent.
But since no genetic material has been found, the protein-only hypothesis is gaining support.
This has been an extremely controversial area of science. Prions are novel infectious agents, and there's been a very rapid evolution of ideas. And I think VSE has arrived on the scene in the midst of this scientific controversy.
The answer is still not known with certainty, but more and more it looks as though the protein-only hypothesis will turn out to be correct.
While the idea continues to be debated, the 1997 Nobel Prize for Medicine was awarded to Stanley Prusner for his pioneering work on prions. What started out as heresy had become mainstream. But now, another issue was taking center stage.
The burning question became, can VSE transmit to humans? And are we going to see an epidemic of human disease following exposure to VSE?
It was an urgent question, and a special scientific team was formed to look for clues that mad cow disease might be passing into the human population in the form of an illness like CJD. But no one knew exactly what they were looking for or what they would find. One member of the team visited CJD patients before they died, looking for any new or unusual form of the disease.
I would travel throughout the whole of the country to see patients with Creutzfeldt-Jakob disease, as the geographical distribution is completely random. Most patients with Creutzfeldt-Jakob disease, by the time that I visit them, are severely demented, so they're unable to communicate. They're usually mute, lying in bed, unable to move. And they have jerking movements of their limbs, what's called myoclonus. They can also, not infrequently, be blind. And even though they can't see, their brain is producing hallucinations, which can make them very frightened.
Another member of the team had the job of examining the brain of anyone suspected of dying of CJD to look for links with mad cow disease. But a CJD autopsy requires special precautions.
I've now changed into the clothes I'll wear to do an autopsy in a case of suspected CJD, and these garments are disposable because they will be incinerated after the autopsy. These are the gloves. On the top is a chain mail handpiece, and this is flexible and allows my hand to be protected from any cuts while the autopsy is being performed. And I wear another pair of rubber gloves on top of this just to make the whole thing as waterproof as possible. And this is the helmet. I'll just put it on. The instruments that I use in the post-mortem room and the instruments the technicians use in our dedicated laboratory really be regarded these as being permanently contaminated, so we use those for CJD cases alone because there is no effective way of guaranteeing decontamination in this disease.
A third member of this new surveillance team was on the lookout for the emergence of any new patterns of CJD cases.
There are a number of possible things we could look for, including a change in the number of cases that might be identified every year. For example, looking at a change in occupation to see whether the people who were in contact with cows or with BSE tissue might be more at risk. And also to look at any change in the age of the patients or the other clinical features. And finally to look at the neuropathology to see if that had changed in any way.
We really had no idea what we were looking for. Our mission statement, if you like, was to identify every case of CJD in Britain and to study the clinical and pathological features to monitor any change.
In the spring of 1995, changes began to emerge. At 19, Stephen Churchill died from what doctors thought was CJD. This puzzling case of CJD in someone so young caught the attention of the surveillance team.
June 1995, we heard about one young patient with CJD, a teenager with CJD. This was clearly very unusual. And then a few months later, we heard about another case in another teenager. And this was clearly exceptional. In December, we began to be referred a number of other younger patients with CJD in their 20s and 30s with a very unusual clinical presentation.
They didn't show the typical appearances, which is in the brainwave recordings that we see in sporadic Creutzfeldt-Jakob disease. This is a brainwave recording from a patient with sporadic Creutzfeldt-Jakob disease, and it shows the classical appearance associated with CJD. These sharp waves are occurring in all across the tracing, coming from the various parts of the brain, and they occur regularly, usually once or twice every second. However, in the new patients, none of them had that classical appearance, and what we saw was just slow waves. They didn't show the typical appearances associated with the sporadic form of the disease.
The first time I saw the new variant of CJD was in a brain biopsy from a young patient, and I can remember being very struck, even in this very small piece of tissue. The changes there were very different from anything that I'd ever seen before.
This was the first confirmed case of a new type of CJD. But what had caused it?
Somebody came down with a questionnaire and asked us questions about lifestyle and eating habits, where we've been on holidays, that sort of thing. Obviously with Stephen being 19, Mum and Dad knew exactly the medical treatments he'd had, his eating, that sort of thing, because he was still living at home at that time. Steve was no different to anybody else. He didn't have any operations that would have put him at risk. He didn't eat anything strange. He was a normal child, and he was a normal 18-year-old up until he became ill.
He did eat beef burgers, but he ate sausages, he ate all sorts of things, no more or no less than most other people. So completely normal in that respect.
Eventually, other cases like Stephen's emerged. And to confirm their suspicion that they had a new form of CJD, The team used a computer to compare brain slides from the randomly occurring or sporadic form of CJD and the new cases. The microscopic holes that accumulate in the brain were outlined in red.
When we first set up the project, we were anticipating that perhaps the changes would be rather subtle. However, when the first new variant cases emerged, the changes were so striking, even on initial examination, that we were overwhelmed by the differences. As well as the spongy change in the tissue, there were large numbers of these plaques, these aggregates of prion protein. But it wasn't just the plaques, they had a particular shape, they were large, they were rounded, and they were surrounded by a ring or a halo of spongiform change, and I had never seen anything like that before. I think I realized then that undoubtedly this was something different, something new, something very disturbing, and something for which we had no explanation.
I think we all had to agree that this was something unexplained and that we couldn't just put it down to a chance occurrence.
By early 1996, 10 cases of the new variant of CJD had appeared throughout Great Britain, all in young people. While there was no hard scientific proof that mad cow disease had caused their deaths, the circumstantial evidence was overwhelming. The news that BSE might have infected the human population shocked the world.
Remember Oprah? She got sued by the beef.
Nations across Europe banned British cattle and meat, just as the United States had done in 1989. Fear struck home when the public learned that the practice of feeding cows the rendered remains of other animals was still legal in the United States.
Cows should not be eating other cows. It has just stopped me cold from eating another burger.
While British beef was still for sale, some restaurants and fast food chains throughout England removed it from their menus. But was mad cow disease really the cause of the new disease?
The timing of these cases is possibly of some importance, and the reason for that is that if the population of the UK were exposed to the BSE agent in the mid-1980s, it would not be unexpected if there were a link that cases would start to occur in the mid-1990s.
The striking similarity in both the clinical features and the pathology in these cases suggests that a common agent is operating, and this, I think, points towards BSE as the most likely cause.
I think the most likely explanation is that these cases have occurred because of BSE.
If these 10 people have died from BSE, then essentially the entire population of Britain is at risk.
But with so few cases, scientists could not accurately predict the extent to which the human population would be affected. In the first year of the cattle epidemic, only 10 cows fell ill. Eventually, the death toll exceeded 170,000 cows. Would the human epidemic follow the same course?
One of the main questions we have to address now is why only this small group of patients has developed this disease. If you believe that this is BSE, then potentially millions of us in Britain have been exposed to the agent in the food chain. So what is special, what is different about these people that, in a way, allowed them to develop the disease?
One of the only clues so far has been found in the genetics of those who fell ill.
There's some evidence from CJD that people of a particular genetic makeup are particularly susceptible to CJD.
The gene responsible for the prion protein seems also to affect our susceptibility to prion diseases. Along this gene, DNA codes for two different types of amino acids, methionine and valine, A person whose DNA codes for one methionine and one valine seems less susceptible to prion diseases. Someone who has two valines is more vulnerable. And those who have two methionines are called methionine homozygotes and seem to be the most susceptible to the new kind of CJD.
The first two published cases were known to be methionine homozygotes, and earlier this year we began to get the results back on the other new variant cases, and this established that there was a link between them, and that was that they were all methionine homozygotes.
All 10 of the new variant CJD cases bore this genetic trait, but this offers little comfort.
Looking at the distribution of methionine homozygosity in the United Kingdom population, it's not really all that reassuring, because about 18 million people in Britain are probably methionine homozygotes.
Testing so many people is out of the question. And scientists are unsure just how much one's genetic type confers protection or vulnerability. In addition, there are still many other mysteries about the risk BSE presents to humans.
It's very hard to try and perform any sort of real risk assessment as to what the outcome of this might be, how many people might get infected, what sort of exposure might be a risk. We really know so little, in fact none really, of the key ingredients that you need to know to make a risk assessment.
For example, no one knows how many British cows contracted BSE,
How much does it take
or
to
how
infect
many
a
entered the
person?
food chain. Complicating risk assessments even further is the mystery of how much infected material a human being would have to eat to trigger disease. Using a herd of test cattle, the British government has established a minimum dose fatal to a cow. Eating just 1/4 of a teaspoon of BSE-infected brain is enough to kill it. But what about humans? It takes a much larger dose to pass infection from one species to another than within the same species. No one knows exactly how much, or whether seemingly insignificant amounts of BSE can accumulate in the body until they reach a fatal level.
We don't know yet whether there's a cumulative dose effect. We know in animal models that there's a minimum dose that you have to give to produce the disease. We don't know yet if you divide that dose up into small portions and give it over a long period of time, whether it would also be effective. And this is an important issue with respect to BSE in humans. It may be that you have to have a very large exposure to BSE over a short period of time to get the disease. It may be that small amounts over years could build up to produce the disease, and we really don't know the answer to that question. But there's likely to be some cumulative element to it, simply because the infectious agent itself is so persistent in the body it can stay there for a long period of time. So it's bound to build up to some extent.
In addition, experts have not determined whether BSE exists throughout an infected animal's body or just in the brain and spinal cord where it accumulates. Flesh, muscle, milk, and blood have never been found to be infectious, although these tests are not definitive. But the greatest mystery is how easily BSE can cross the species barrier from cows to humans. A strong species barrier would protect most people, but a weak one would leave many more susceptible.
The species barrier between cows and humans was unknown. You can't just assume that it's going to be an absolute one. Of course, we can't measure it because that would involve injecting humans with BSE, which clearly we wouldn't do.
Scientists need to know more about what controls the species barrier.
And the only way to find out is to do the experiments. You can't just sit down with a piece of paper and work out how easy it will go from one species to another. It's something you have to determine experimentally.
One of those experiments focuses on how the species barrier works on a molecular level.
Evidence from other labs has suggested that the type of Prp protein that an animal makes can determine whether or not that animal is going to be susceptible to infection with the agent from a different species. So what we know is that this protein, this PRP protein, it's composed of a sequence of what are called amino acids, which are linked in a long chain. And that the amino acid sequence of PRP from an animal like a sheep differs from the sequence in an animal such as a cow. And what we're interested in determining is if it's those differences between these two types of PRP that can affect whether or not this species barrier is broken and infection can occur.
Priola is investigating how scrapie is passed between two closely related animals, hamsters and mice. Curiously, mice can't be infected with hamster scrapie, but hamsters can be infected with mouse scrapie. To understand why, Priola analyzed the differences in their PRP proteins.
Between the mouse PRP and the hamster PRP, there are 16 amino acid differences. And what we were able to do is show that you can control formation of this protein by changing just one amino acid. So one change out of 254 amino acids could be enough to allow the formation of this abnormal protein. If you get the formation of this abnormal protein, you're probably going to get disease. One of the things that this piece of work implies is that very minor changes in the PRP proteins between two different species could really have a dramatic effect on whether or not you get formation of this abnormal protein.
This work indicates that the species barrier against BSE can be breached fairly easily, which would seem to have grave implications for humans. Experiment after experiment is confirming that this is already happening, leaving little doubt that new variant CJD is in fact the human form of mad cow disease. To date, it has killed more than 20 British people. So far, the United States has been lucky. No cases of BSE or new variant CJD have ever been found here. But can it remain that way?
Yes.
I think the likelihood of BSE existing in this country is small. It's not zero, but it's small. And I think the phenomenon that BSE is widespread in this country is simply not true.
To prevent BSE spreading in the food chain, the United States government has at last banned the practice of feeding most, although not all, animal remains to cows and sheep.
Bone meal.
Thanks to similar measures in England, BSE is now on the decrease there, and may even be eradicated from British cattle herds early in the next century. But it remains to be seen what direction the human disease will take.
It's far, far too early to say what sort of epidemic that's going to be, whether it's just going to be 20 or 30 cases over several years, or whether it's going to be hundreds or potentially thousands of cases. We have to wait and see.
If the 24 cases of CJD in Great Britain are most of the cases that we will ever see, that is to say, if what has happened basically represents a few susceptible people, then I think even if BSE existed in this country, it would not be especially panic-producing. If, on the other hand, within 2 or 3 years, we start to see hundreds or thousands of cases of CJD in humans in Great Britain, then that's going to be cause for concern all over the world.
Okie dokie. So this is an example of something where finally we get things a little bit right in the fact that they were able to stop the epidemic and everybody knows about it now. And there's major efforts to contain it, although in this country we don't test cows for it as much as other places. But still, less than 3,000 people worldwide, and no significant new cases in England since they instituted the control. Okay, so now we're going to get the test back. Sadly, I had one of the student assistants run the Scantrons, and they ran the wrong side. So if you see a piece of tape, that means they ran the wrong side. I redid it. I redid them all, which is why I was up till 4 in the morning. They are right. The number in green is the right one. The circled number percent in green is the right one. And it includes the adding of all your word questions. So when you get it, just say, I'm here, and come and get it, and walk quietly out. No sobbing, no jumping off of bridges. Remember that the 25 points that you're going to do for the parasites are going to bring this up. So anybody that made in the middle 60s to the, well, if you made an A, a B, or a high C, you can still get an A in the class. If you made a low C and or a high D, or even like a 59, you can still get a B easily into the class with the bonus and a good paper. So no suicides, please. Flores, Bromberg.
Here.
Sorry, I wrote over your names. You might read them. Rizzo, we're done for the day. So don't forget Friday, if you can come for that. We're going to hand out some papers for Rizzo. On, A-N. Karen. Guillermo.
Oh, here.
Hanvin. Lydia. Lydia here. Solomon. Amelia. Susanna. Susanna made one of the hundreds. Yeah. Kim. Natalia. Handler. Also made one of the hundreds. Thank God you got all the word questions. I mean, no, I'm not making a comment, but let's just put it this way. If it wasn't for the word questions, we would all be crying and committing suicide. Daryl? Nguyen? The green one is the right number. Katoon?
Okay.
Which is why she doesn't do it. Never shows the class. Gonzalez?
The green. That's your side.
Amy. Irina. Watson. Laura. Perez. P-R-E-Z. Anyone who didn't get there? Last name? Yeah.
I got a 93. That's awesome. 100. Did she get all the points? You got all of these right? I got 13. 13. I'm not terrible.
Did you have the blue test or did you have the white? I had the blue test. I had the blue ones. Okay. Is that got taken out? Oh, yeah. Okay.
Yeah, hit the off with the silver button. That's all right.
This one?
Yeah, thank you.